This is a brief introduction to Sequencher connections, added to Sequencher with version 5.2. Sequencher connections is a response to a need of many of our clients to take lots of data they're generating and aligning with Sequencher and then apply multiple different tools to them. Not necessarily in a pipeline, but just different tools that are used that could be done in parallel and using different resources on the computer, on the web, and elsewhere. I'm going to open up a simple sample project here. This is reason number one. I could take all of this data or any small portion. I'll just pick a few to show how this process works. This is just like any other Sequencher project that you might be familiar with. And I can take, let's say these first three, open them each in a connections window. So I can. There are certain kinds of analyses that you perform on individual sequences, and other analyses that you perform on groups of sequences. For instance, you can do an alignment on a group of sequences, but makes no sense to do an alignment on a single sequence. So here's something I'm going to do on several sequences. I've got, by default, a primer blast, a blast against the NR database at NCBI. I'm going to add a few channels. Say I had 2 or 3 or 4. Don't want to make this too lengthy. I'll make this a little wider so I can see what's going on. And I won't do the same kind of analysis, but maybe I want to, let's say, run against the human EST database. Let's come up with kind of a nice human color there. Maybe the mouse DNA database. Something great for a mouse. And let's say let's take RNA and do things a little bit different. We'll filter for human repeats and use regular blast and set the board size down to 15. And what shall we make this one? Let's see what's being done in the high throughput group today. Now, I've set up these parameters. I can run all of these sequences at once and say run these sequences against all these different tools. I'll just narrow this down a little bit for primers. I won't do this for all of them, but let's say I'm interested in a particular short range, let's say between 700, say 700 and 900. I can say run all. And data is being sent off, in this case, to primer blast, to the NCBI blast site. Let me make these a little more even. So when it was sending, the data was being sent off to NCBI. Pending, we're waiting for results to come back. You can see we have a full web browser down here. I can look at any one of these sequences. If I want to look at these results, here's the NCBI version of the blast parameters I received back. This is the sequence I sent off. Since I'm looking for primers, I can get primer descriptions this way. And you can see some of these are coming back as we go. So let's take a look at this. Here's the sequence against the EST, the human EST database. I'll look at NCBI's view. Here's the same sequence against the mouse database. And you've all seen these before, and you have to kind of take a look at them individually. One of the things that we can do here is, say, put these all together in one schematic. Move this onto the screen for you. And so here is the same sequence run against the HTGS database, the RefSeq database, the mouse and the human database. I can look at any one of these in several ways. So if I'm interested, maybe I found an unexpected match here, I could right-click or control-click on an Apple keyboard and open this in a new window. because again, this is a full function browser. I can say I'm interested in this and actually add this one back to, or any one of these back to connections. This one is now a new tool that I can run. I can run these individually. So individual channels or all of them at once. I can cancel them as I go. Without going into too much more detail, there are different flavors of connections tables, 2 to start with and more to come. So I can let this run. This is using resources that are off your computer. This is going off and collecting information from BLAST. I could just as easily, if I wanted to, make one of these channels a local BLAST, if I have local BLAST installed on my machine. And there are other options, as you might imagine. But let's say instead of looking at individual sequences, I have a group of sequences. I'll pick an easy example. Here's just a bunch of HLA types. And I can say pick some of these, or since I'm doing a multiple sequence alignment, I'll do all of them. And instead of opening each in a connections window, I'll open them as a group. And I'll call that group 83 HLA profiles for demo. Now in this case, I have one group with that contains 83 sequences. I set it up so that in one case I am not using the diagonals to speed up. I'm doing 5 iterations through the muscle algorithm. And the others I'm going to go quicker. I'm going to do only 2 iterations and use the diagonals to speed them up. Let's see what kind of differences I get. So I can say run them all. As you expect, this one finished first. If I click on this cell, I can see the HTML version of the alignment. I'm playing text alignment for those who need to use it. The log files are there for people who are more expert in algorithms like muscle. And the phylogenetic tree that people have asked us for a while. And we'll be adding to this quite a bit over the next few weeks and months. But different formats of trees. I can, of course, do the obvious things and zoom in and out. I can change it to different formats. And you can compare the results between the 2 settings to see if things change at all. All right, that's just a quick introduction. We look forward to your feedback. Thanks very much.